Hello everyone and welcome to the biology lecture series. And today we'll be discussing the topic sexual reproduction in the flowering plants. So this is the continuation of the previous lecture. So in the previous lecture we were studying about the pollination. So I've told you what is pollination. Pollination is the transfer of the pollens from the anther to the stigma. Okay, this is known as pollination. Now pollination we have seen it is of three types: autogamy, tonogamy and xenogamy. There are three kinds of pollination, and we have studied what is the difference between three kinds of this pollination. Okay, first is autogamy, second is tonogamy, and the third one is the xenogamy, which is the actual cross pollination. Now, in this lecture, we'll continue with the pollination. Okay, so whenever the pollen lands on the stigma, then it is compatible or it can be incompatible. So if it is compatible, then it will germinate and it will form the pollen tubes. All right. So now, if it is of right type, means if the pollen is of right type, means if it is compatible. Right type means if the pollen is compatible. So here what we are studying about the pistil and the pollen interaction. What we are studying about the pistil, which is the female reproductive part. Okay, and we are talking about the pollen, which is the male gametophyte. We are talking about the interaction between them. Okay, so pistil, what are the part of the pistil? It is ovary, style and stigma. Now, if the pollen is of compatible means, pollen is of right type means if it is compatible, then the pistil accepts the pollen and promotes the post-pollination events that leads to fertilization. Okay, so first step, we were talking about the pollination. Now we are talking about the events that are happening post-pollination. Clear? So first of all, the pollen should be compatible and it should be accepted by the stigma, which is the part of the pistil. Clear? Now, if the pollen is of wrong type, means if it is incompatible. If it is incompatible, the pistil will reject the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. Okay, so let's see if the pollen is incompatible. Then what will happen? Either the pollen will not germinate, either the pollen will not germinate, or it will germinate but the pollen tube, okay, or you can say the pollen tube, okay. So pollen germinates to form the pollen tube. If this is the pollen, it forms the pollen tube. All right, so what will happen? Either the pollen tube growth will be blocked in the style. Okay. Style. So you know, whenever the pollen germinates, the pollen tube, this travels through the style. And the pollen tube, it carries what? It carries the two male gametes. So there are two male gametes here. Okay, clear? So in either of these cases, what will happen? There will be no fertilization. Okay, so either of these cases, there will be no fertilization because the male gametes, they do not enter the embryo sac where the egg is present. Okay, clear? Now, Next point is the ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between the pollen grain as well as the pistil. So there are a lot of interaction that are happening at the pistil, between the pistil, between the stigma of the pistil as well as the pollen grains. And the pollen's acceptance or rejection, this is based on what? This is based on the interaction between the pistil as well as the pollen grain. Now next, this dialogue is mediated by the chemical components of the pollen interacting with those of the pistil. Clear? So some chemicals are involved here. Okay, so there is an interaction between the pollen and the pistil. How is this mediated? These are mediated by the chemical components of the pollen grain which interact with those of the stigma of the pistil. Now what are these events? Okay, so we can write what are these. These are the how the interaction between the pollen and the pistil takes place. We'll be studying about this. All right. So you can write here what we are studying. We are studying about the pollen and the pistil interaction. Okay. So first of all, pollen proteins. Let's talk about the pollen proteins. Now, pollen grains contain various proteins, including recognition proteins that help the pollen identify the compatible pistils. Okay, clear? Let's read it again. Pollen grains contain various proteins, including the recognition proteins. So some recognition proteins are present in these pollen grains that help the pollen to identify the compatible pistils. Now let's talk about some of the receptors that are present in the stigma. Now the stigma, which is the receptive part of the pistil, it contains specific receptors that recognize and bind to this pollen proteins, okay, or molecules present on the surface of the pollen grains. Now this recognition triggers further biochemical reaction. So what is happening here? Now the pollen contains some pollen proteins. So these pollen proteins, these are recognized by the receptors present on the stigma. Okay. And whenever these pollen proteins are recognized by the receptors, then a biochemical cascade is triggered, or biochemical, you can say, reactions are triggered. So biochemical reactions, those reactions that involve enzymes. Now let's talk about the pollen-pistil interaction. The pollination does not guarantee the transfer of the right type of the pollen, means the compatible pollen of the same species as the stigma. What do you mean by right type of pollen? Right type of pollen means compatible pollen of the same species as the stigma. Okay. Now often the pollen of the wrong type, either from other species or from the same plant, which is known as self-incompatible, also land on the stigma. Okay. Now the pistil has the ability to recognize the pollen. Okay. Because pistil has what? Pistil has receptors that recognize the pollen proteins. Okay, whether it is of right type, means compatible, or it is of wrong type, which is known as incompatible pollen. Now, as mentioned earlier, following compatible pollination, so if the pollen is compatible, or if the pollination is compatible, then what will happen? The pollen grain germinates on the stigma to produce the pollen tube. Okay, through one of the germ pores. So through the germ, so pollen has a germ pore, and the pollen tube, it arises through the germ pore. Okay, clear? Now this is an image. This is an image showing you the pollen grains that are germinating on the stigma. Okay, clear? Stigma is the part of the pistil. You can see here, these are the pollens that are germinating on the stigma. Now the contents of the pollen grain move into the pollen tube. So what are the contents of the pollen grain? Male gametes. Male gametes, it moves into the pollen tube. So if you know about the structure of the pollen, usually the pollens, I've told you earlier also, they are released in two cell stage. One is, okay, so we are talking about the two cell stage pollen. Now this is the vegetative cell and this is the generative cell. Clear? Vegetative cell is large and generative cell is small. Okay. So two cell stage. So generative cell further divides to give rise to two male gametes. Divides to form two male gametes. Okay. So this is known as three cell stage. Okay. This is two cell stage. Now let's talk about the three celled stage. So in three cell stage, what cells are present? One vegetative cell and the two male gametes are also present. So three cells are present. So this is known as pollens at the three cell stage. So the contents of the pollen, especially these two male gametes, these are released in the pollen tube. Okay, so this is the pollen. Okay, so these are the pollen, these are the male gametes. So the contents of the pollen, these are released in the pollen tube. Okay, this is what? This is the pollen tube. Now the pollen tube grows through the tissue of the style and the stigma and the style and reaches the ovary. Clear? So pollen tube, it will travel through this stigma as well as the style and it will reach the ovary. Now within the ovary, what is present? Ovule is present. And ovule, it consists of the embryo sac. Okay. Then the pollen tube, it will enter into the embryo sac. Okay, through the micropylar region of the embryo sac. Okay, so you can write here the ovary. Okay, it consists of the ovule, or you can say embryo sac is present within the ovule and ovule is present within the ovary. Okay, now this embryo sac, pollen tubes finally enter into the embryo sac. It will enter into the ovule and then it will enter into the embryo sac through the micropylar end. So you can write here the pollen tube enters into the embryo sac from the micropylar end. Okay, we'll talk about this when we'll talk about double fertilization. For now, you have to remember that the pollen tube enters into the embryo sac through the micropylar end, and embryo sac contains what? It contains the egg cell, and then the male gamete will fuse with the egg cell, and this process is known as fertilization. Clear? Now this is an image. Image B. What is image B? Here the pollen tubes growing through the style. So these are the pollen tubes that are growing through the style. And figure C, this is the LS of the pistil, means lateral section of the pistil showing path of the pollen tube growth. Okay, so what are these? These are the pollens. So these pollens, whenever, if they are compatible, okay, and if they are recognized by the receptors present on the surface of the stigma, then what happens? They will germinate and they will produce the pollen tube. So this is the pollen tube you can see. And the pollen tubes you can see, it is traveling through the style. So this is style. This is the stigma. Okay, this is style. And then the pollen tubes, it will enter into the embryo sac finally. You can see here that pathway of the pollen tube. It is traveling. And then this is the embryo sac here. So embryo sac consists of the antipodals, polar nuclei, axil as well as the synergid. So these are the cells of the embryo sac. Embryo sac is also known as the female gametophyte and it is present within the ovule. Okay. So it is known as the female gametophyte. Clear? Now the embryo sac consists of the egg cell, okay, which will be fertilized by the male gametes, which are derived by the pollen tube. So this is a longitudinal section of a flower showing the growth of the pollen tube. Clear? And the pollen tube, it enters through the micropylar end. So this is the micropylar end. Okay, and the other one is known as the chalazal end. So at the micropylar end, the egg apparatus is present. So egg apparatus consists of this egg cell as well as the synergid. And this is the chalazal end where the antipodals are present. Clear? So this one will be the chalazal end. Okay, clear? Now let's move on. Now you would recall that in some plants, the pollen grains are shed at two cells condition. So a two cell condition means one vegetative cell and one generative cell. And then in such plants, the generative cell divides and forms the two male gametes. So when the generative cell divides and it forms two male gametes, okay, then these two male gametes, they are transferred through the pollen tubes. Now in plants which shed pollens in the three cell conditions, what happens? The pollen tube carries the two male gametes from the beginning only. Okay, so I've already told you two cell condition means one vegetative cell and the generative cell divides to form two male gametes, and the two-celled embryo pollen grain means one vegetative cell and one generative cell. So here, now the pollen tube after reaching the ovary enters into the ovule. Okay, I've told you ovule is present within the ovary through the micropyle, through the micropylar end, and enters one of the synergid through the filiform apparatus. Okay, so what is happening here? Let's draw the embryo sac here. Okay, so this is a very rough diagram that I am drawing. So the embryo sac consists of egg apparatus, antipodals, and the polar nuclei. So suppose this is the micropylar end. Okay, at the micropylar end, egg cell is present and these are the synergids. Okay, this large one is the egg cell and these two are the synergids. Now the synergids consist of the filiform apparatus, which I am drawing in the red. So these are the filiform apparatus present in the synergids. So this is the synergid. Okay. So if this is the synergid cell, it consists of this filiform apparatus. So what is the function of this filiform apparatus? This is filiform apparatus. And this is what? This is the synergid. Now, this what is the function of this filiform apparatus? It guides the pollen tube. So let's write it first. It guides the pollen tube entry. Okay, so it guides the pollen tube, and the pollen tube, it enters one of the synergids. Okay, it will enter through one of the whenever it enters the embryo sac, it enters through this synergid. And what is the role of this filiform apparatus? It guides the pollen tube entry into the embryo sac. Okay, this is the function of the filiform apparatus, and the pollen tube enters through one of the synergid. Okay, and then it will release its contents, that are the two male gametes. Clear? So this egg cell and two synergids. So two synergids are present. This is known as the egg apparatus. Then we have the two polar nuclei in the center. These are the polar nuclei, and then we have these are the antipodals, which are present at the chalazal end. Clear? What is this? This is the embryo sac. It is also known as the female gametophyte. This embryo sac is present within the ovule. Clear? So now it's clear how pollen tubes enter into the embryo sac. It enters through one of the synergid, and the filiform apparatus, they guide the entry of the pollen tube into the embryo sac. And then when the pollen tube enters into the embryo sac, then it releases its two male gametes. Now this figure D, you can see what is this D? This is the enlarged view of an egg apparatus, showing the entry of the pollen tube into a synergid. So you can see this pollen tube, this is entering into the one of the synergids, okay, which is present within the embryo sac. Here are the two male gametes, and this is the vegetative nucleus. All right, this is the filiform apparatus. So filiform apparatus guides this pollen tube. This is the synergid cell. Okay, this is the central cell within which the two polar nuclei lies. This is the egg cell. Okay, and all these cells, these are haploid. Now image E, what is image E? This is the discharge of the male gamete into the synergid and the movement of the sperms. Okay, one into the egg, another into the central cell. So male gametes are released inside the inside the embryo sac. Or you can say the male gametes are released where? Into the synergids, and one of the egg cell will fertilize with the. Okay, so there are two male gametes. So we will discuss this in detail when we will talk about double fertilization. So there are two male gametes, one male gamete. This will fuse with the egg, and the other male gamete, the second male gamete, this will fuse with what? It will fuse with the two polar nuclei. Clear? This is known as double fertilization. We'll talk about this in the next lecture. Now, many recent studies have shown that the filiform apparatus present at the micropylar part of the synergid guides the entry of the pollen tube. Okay, so this is the function of the filiform apparatus. Now, all these events from pollen deposition on the stigma until the pollen enters into the ovule and finally into the embryo sac are together referred to as pollen-pistil interaction. So finally, how can you define pollen-pistil interaction? Okay, what is the first step? Pollen deposition. Okay, so pollen deposition. Okay, then pollen tube formation. Okay, then third one is the entry of the pollen tube. Entry of the pollen tube into the ovule, or you can say into the embryo sac finally. Clear? So these are together known as the pollen-pistil interaction. Clear? Now, as pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition. So first of all, what was the first step? Pollen recognition by the receptors present on the stigma, followed by promotion or inhibition of the pollen. Means, if the pollen is compatible or if the pollen is recognized by the receptors, then the pollen will germinate. Otherwise, the pollen growth will be inhibited. Either the pollen won't germinate or the pollen tube growth will be blocked in this style. Now, the knowledge gained in this area would help the plant breeder in manipulating the pollen-pistil interaction, even in incompatible pollinations, to get the desired hybrids. So, in order to make the hybrids, so if the knowledge of this pollen-pistil interaction is there, then a plant breeder can utilize that information to produce hybrids even if the pollen is incompatible. Now, you can easily study pollen germination by dusting some pollens from the flowers such as pea, chickpea, crotalaria, balsam, and vinca on a glass side containing a drop of sugar solution, about 10% sugar solution. What is the concentration of sugar solution? It is 10%. Okay, so you can just dust the pollen grains. Okay, you can bring the flower of these plant and you can dust this flower. Okay, or you can dust the pollens from this flower on the glass slide that is containing a drop of this sugar solution. Now you'll observe after 15 to 30 minutes, if you observe this slide under the low power lens microscope, then you can see the germination of the pollen tubes. Okay, clear? You can see the pollen tubes coming out of the pollen. Now, a breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior varieties. Clear? This is known as artificial hybridization. Okay, artificial hybridization is a man-made thing. Alright, it is done to produce the hybrids. So we select the desired trait and we want that traits to be in the hybrid. But what happens due to pollen incompatibility it is very difficult to cross them. Okay, so if we know about the interaction of the pollen-pistil interaction, if we have the knowledge of the pollen-pistil interaction, then we can easily do this hybridization process. This artificial hybridization is the major approach for the crop improvement because we are selecting the desired characteristic here. Okay, and this leads to the increase in the yield as well as the vigor of the plant. Okay, so plant can be stress tolerant. Okay, abiotic stress tolerance can be there, or biotic stress tolerance, or we can introduce the genes. We can not introduce, we can select that gene. Okay, that increases the yield. Okay, now next, artificial hybridization. What is artificial hybridization? It refers to a controlled breeding process conducted by the humans to cross-pollinate or cross-breed two different plants, often of the same species but different, sometimes of the different species, to create offerings with the desirable traits. Now, in such crossing experiment, it is important to make sure that only the desired pollen grains are used for the pollination. So, desired pollen grain which contains the desired characteristic. And the stigma is protected from the contamination from unwanted plants. Okay, so this is very important. Now, this is achieved by emasculation. Emasculation means removing of the anthers. And another one is the bagging techniques. Okay, so contamination from the unwanted pollen is avoided by emasculation. What is emasculation? It is the removal of the anthers. So if the female parent bears the bisexual flowers means the flowers that contain both the male and the female reproductive system. Removal of the anthers from the flower bud before the anther dies using a pair of forceps is necessary, and this process is known as emasculation. Now, the emasculated flowers means where the anthers has been removed had to be covered with the bag of suitable size, generally made up of butter paper, to prevent the contamination of its stigma with the unwanted pollen. So, flowers are open and the stigma and the anthers, they are exposed to the external environment. So, what can happen? Any foreign pollen can land on the stigma. Okay. So, what we have done emasculation because we want the pollination to be done by the pollen that we have selected that has all the desired characteristic. Okay. So, we have removed the anther. Okay. Then we don't want any unwanted pollen to land on the stigma. So that's why we do the bagging. Clear? Now this process is known as bagging, in which we use the butter paper, okay, to bag the flower. Now when the stigma of the bagged flower attains receptivity, the mature pollen grains, okay, that contains all the desired characteristic or the desired traits that we want from the anthers of the male parent are dusted on the stigma, and the flowers are rebagged and fruits are allowed to develop. Means then the fertilization will take place. Okay, and after fertilization, you will get the seeds as well as the fruits. Now, if the female parent produces unisexual flowers, then in this condition, what happens? There is no need of emasculation. Okay, because what are unisexual flowers? They consist of either the male or the female reproductive part. So either it will contain stamens or it will contain the pistil. Now the female flower buds are bagged before the flowers open, okay, to prevent the pollination from the unwanted pollen. Now when the stigma becomes receptive, the pollination is carried out using the desired pollen and the flowers are rebagged. So there are two cases. First, whenever the plant produces bisexual flowers, in that case you have to do emasculation as well as the bagging. Whenever the flower produces unisexual flowers, then there is no need of emasculation. You can only do the bagging, and then you have to pollinate it with desired pollen, and then you have to rebag it. All right, clear? So I hope you enjoyed this session. All right, we'll discuss about the double fertilization in the next session. Okay, thank you very much for listening.